Hello. I will talk on how our targeted proteomics study corroborates the subtypes of LUTS that were identified using symptoms data. This is my disclosure slide indicating that I have nothing to disclose other than being involved in two NIDDK funded projects that provided data and foundation for my today presentation. The first project is Symptoms of Low Urinary Tract Dysfunction Research Network, or LURN. The goals of LURN are to improve measurements of LUTS, identify subtypes of LUTS, and collect biosamples for future research. In the second project, we performed targeted proteomics of serum in people with LUTS by using proximity extended assay by Olink Proteomics. In particular, we used three panels: cardiometabolic, inflammation, and neurology, and measured abundances of 276 proteins. As far as we know, this is the first large-scale targeted proteomic study of LUTS. In this presentation, I will be talking about 230 baseline serum samples from women with LUTS in comparison with 30 non-LUTS controls that are frequently matched with cases by sex, age, race, and BMI. We believe that LUTS, as many other complex disorders, is heterogeneous and could be represented better as a set of subtypes. Our hypothesis is that subtypes of women with LUTS identified using symptoms, physical exam, and demographics data will be biochemically distinct and will demonstrate different signatures of over and underabundant proteins. Therefore, we will first identify subtypes of women with LUTS without using biomarker data, and then compare biomarker signatures of the identified subtypes. Here's the list of 185 variables used for clustering. Urologic symptoms were measured by LUTS Tool and AUASI. We also used a bunch of demographic, physical exam, and comorbidities variables, as well as 5 bladder diary variables. We used a resampling-based consensus clustering algorithm, which resulted in identification of 5 distinct clusters represented by these radar plots. The upper row shows urinary symptom signatures that are color-coded. For instance, frequency is coded green, while incontinence is yellow. The second row represents the clinical variables and demographic data. Here's the brief verbal description of the identified clusters. All of them demonstrated high urinary frequency, and none could be characterized by a single symptom. Cluster W1 presents minimal incontinence and mild voiding symptoms. W2, mild urinary symptoms and significant prolapse. W3, high frequency urgency and urgency incontinence. W4, multiple symptoms associated with stress incontinence. W5, high severity of LUTS for all symptoms. This is the slide for those interested in cluster analysis. It shows how we determine the number of clusters and made clear that identified clusters are distinct. The methodological details of our clustering procedure are available in bioarchive. We used Olink assay to measure abundances of 276 proteins in serum of 230 women with LUTS and compared them with 30 controls. The first volcano plot demonstrates the presence of multiple significantly different overabundant and underabundant proteins in all 230 women with LUTS relative to controls. And the rest of the plots do the same thing for each of the identified clusters. While some of the differential abundant proteins have been associated with LUTS previously, such as TNF, IL-10, MSP, TGF, the majority of the proteins are novel. One can make several interesting observations from the volcano plot. The highest number of differential abundant proteins are in cluster W5, which demonstrated the highest level of urinary symptoms. Overlap between the lists of differentially abundant proteins is low, meaning that clusters are biochemically different. Highest overlap is between clusters W3 and 5, both characterized by high frequency, urgency, and urgency incontinence. Lowest number of differentially abundant proteins are in clusters W2 and 4, which are driven by anatomic rather than biochemical factors. Mapping of the differential abundant proteins on the known biochemical pathways with Metaco software demonstrated that affected pathways are different across the clusters as well, confirming that they are biochemically distinct and likely have different etiologies. For instance, there is no overlap in the top 10 affected pathways for clusters W1 and 3. In conclusion, 5 distinct subtypes of women with LUTS were identified by clustering urologic and non-urologic PROs, demographics, and physical exam data. Targeted proteomics study of serum in women with LUTS identified multiple differential abundant proteins. Affected proteins and biochemical pathways are different across subtypes of women with LUTS, corroborating that identified subtypes are biochemically different. Since proteomics data were not used for the clustering, this result independently corroborates the distinctiveness of identified subtypes. And of course, further investigation of the etiologies and clinical significance of the identified clusters are still necessary. Thank you for your attention. I am happy to take questions.